Hey guys, my name is Catie Gutierrez. Welcome to our presentation of P1Pk in the Globoside systems. I will be going over the objectives and then going through the P1 antigen and antibodies, and then Chandler will take over for the PK and the Globoside systems. These objectives are the ones that Justin posted on our Canvas modules. I just put these on here so we're all on the same page and we know what we're looking for as we go through the presentation. So I'll just quickly read through these for you. So we're going to focus on describing properties of the P1Pk and Globoside systems in terms of clinically significant alloantibodies, immunogenicity of the antigen, antibody specificity, antigenic dose and frequency, number and location site on the cell, modes of inheritance and development, known biological function of antigen, associated disease states. We're going to describe the antigens present, phenotype frequency, and antibodies produced in the following phenotypes: are P1, P2, P0, P1K, P2K, and list the known antigens within both of the P1Pk and Globoside systems. List sources of P1 substance and describe how P2 individuals can form anti-P1 antibodies upon exposure to these substances. Recall the source of P1 substance that can be used to neutralize anti-P1 antibodies in vitro. We're going to describe anti-P1Pk in terms of clinical significance, antibody isotype, form or name, which phenotype produces antibody specificity, thermal range activity, complement binding, implications in severe hemolytic transfusion reactions, and hemolytic disease of the fetus and newborn. We're going to state the autoantibody specificity that is most often involved in paroxysmal cold hemoglobinuria, describe the condition and describe the test used to confirm it, define the term thermal amplitude, and describe how this applies to determining the clinical significance of cold reactive antibodies. So we're going to start out with our P1Pk system. This is just made up of our two antigens, our P1 and our Pk. So legitimately what you see in the name is what is in there. Just a side note, it does not include the P antigen. That is part of the Globoside system. So really just P1Pk, that's a commonly missed question. The ISBT number is 003. It is located on chromosome 22. They are synthesized by glycosyltransferases on type 2 H chains. So those are the precursor chains that also are used for like our ABO and RH typing and stuff like that. We can find these antigens on red blood cells, lymphocytes, granulocytes, or monocytes. But interestingly enough, even though they use those H chains, they have not been detected or identified in secretions. So all antigens in the P1Pk system are enhanced by routine enzymes like papain, trypsin, and bromelain. So just remember Pickler, and that P stands for the P system. So onto our P1 antigens. The expression of these P1 antigens can start at roughly 12 weeks of gestation. You can find them on the fetus red blood cells. However, as the fetus grows, these antigen expressions weaken, and they stay weak up until about 7 years of age, and that's when they're fully expressed. But even after that, at full expression, they are highly variable, meaning one person might be a strong expressor and another may be a weak expressor, and this could be due to genetics, or it may be due to homozygous versus heterozygous inheritance. It also can be dependent upon race. 79% of Caucasians are positive for the P1 antigen, and 94% of African Americans are positive for the P1 antigens. So again, it's more likely that those African Americans will be strong expressors of this P1 antigen. Even though this P1 antigen may be more frequently found or encountered, it is actually a weak antigen. It degrades easily and rapidly in saline. Another aspect to it, it's the only antigen. There's no opposite partner or antithetical partner to this. It's not like M and N or big S and little s. It's more similar to our RH antigen where you're either positive for it and you have it, or you're negative and you don't have it. So in regards to that, you either are P1 positive or you're P1 negative. But I also wanted to note that P1 negative can also be denoted or known to be called P2. So if you hear P2, just know that means P1 negative. So onto our anti-P1 antibody. Obviously, when individuals are antigen P1 negative, they can form anti-P1 antibodies, mostly alloantibodies. So some of the characteristics of these antibodies, they are a weak IgM. They are non-RBC immune, meaning you can be exposed to them through non-blood products. Anti-P1, again, is relatively common but rarely seen in lab testing. This is due to the immediate spin phase often being skipped during testing. Just because, if we're looking for warm antibodies because they're more clinically significant, obviously they're most likely to skip the immediate spin phase. But I do want to point out, very extremely rarely, you will see these anti-P1 antibodies react at the incubation phase or the AHD phase. They can, and in your IAT you'll see them bind complement, and when they do this, it is good to note that they can cause hemolytic transfusion reactions. Again, very rare, but just know if you do see them, they can cause those reactions. And then also on your screening cells, you'll probably notice the irregularity of the reactivity. So with screening cell 1, you might notice a 1+. With screening cell 2, it might be a 4+. Screening cell 3, again a 1+. Just due to the irregularity of expression of these antigens, you're going to get that irregularity seen across your panels, so be aware of that. And again, like we already talked about, it's IgM, it's a cold reactive. And when we think of cold reactive, we normally think room temp, but in our textbook, this actually states that it prefers to react a little bit colder, more around 4 degrees Celsius, so it really does like the cold. So let's go back to our non-RBC immune part of this. How and why do we form non-RBC immune anti-P1 antibodies? So there are a few ways that we can do that. One would be exposure to hydatid cyst fluid. These cysts are formed by infection with the Echinococcus granulosus tapeworm. These things are nasty. I wish I would have included a picture of these cysts, but they are found, like sheep farmers or sheep herders are known to be infected with this tapeworm. That's really common in sheep and around sheep. So we actually have quite a few of farmers and herders here in Utah, so it wouldn't be uncommon to see anti-P1 antibodies here in Utah. They are also found in pigeon and turtle dove droppings. And then also we can use the egg albumin of these turtle doves and pigeons to neutralize the anti-P1 antibody in vitro, which is really neat. I found this chart and thought it would be a good tool to use for a quick review of our P1 antigen information that we've gone over. So our ISBT system symbol or name is P1Pk. ISBT system number is 003. Clinical significance is no. Asterisk unless seen at the 37 degrees phase, and then just make sure you know what type of reactions you'll see. Antibody class is IgM. Optimal temperature is room temp, and even down to 4 degrees Celsius. Again, it likes it super cold. And then our reactivity of the antibodies can be enhanced with enzymes. That is all I have for you guys right now. Chandler's going to take over for us, and he's gonna go over the PK, the P, and the rest of the Globoside system for you. So thanks so much.
All right, thank you, Catie. So moving on to P and PK antigens. Both are considered high incidence antigens, and 100% of individuals are P positive and PK positive. Something else I found is that both have wide tissue distribution and has been found to be a receptor to many pathogens and toxins. Let's talk about P0, also known as little p phenotype. To have the little p phenotype, you must lack the big P, big P1, and PK antigens. If somebody is the phenotype, if someone is the little p phenotype, then they will produce an antibody called the anti-P1Pk antibody, which in turn it is just 3 different antibodies, but they call it anti-P1Pk. And it is highly hemolytic. And this part I want to come back to. Let's talk more about anti-P1Pk. It shares both IgM and IgG antibody classes that can react over a wide thermal range and bind complement very efficiently. This antibody is in all individuals with little p phenotype. Anti-P1Pk will react with other red blood cells except other red blood cells of the little p phenotype. So why would this be considered a problem? And so with that said, I want to go back one slide. So a recent case in India showed that the gene that codes P1 and PK, A4 GALT, had a deletion in the gene that was the cause of the P0 phenotype. In this medical paper, it talks about an individual in India who got into a car crash and they needed to have blood. And so it came to find out that he was P0 or little p phenotype. So if this person was in a car crash and he needed blood, what type of blood would he get? Let's say for myself, since I more than likely have the big P, if my negative O blood were to be given to him, red blood cells, as a blood transfusion, there would be a strong hemolytic reaction. The antibodies in his plasma would be reacting to my P antigens on my red blood cells, and it would be causing hemolysis. And then also trying to find someone else with a little p phenotype to be able to donate him blood and transfuse it is very difficult because the little p phenotype is very rare because P and P1, because P and PK, remember, are very high incidence antigens. So then they would have to go get on a red blood donor list, and that can make things difficult. Now let's talk about allo-anti-P. It is produced by individuals with the PK and little p phenotype. So little p is obviously already going to make it because it's making all the antigens against it, but also big PK can also produce the allo-anti-P. Not to be confused with P1, which is a different antibody produced by individuals who lack the P1 antigen. Now let's talk about auto-anti-P. It is mostly found in young children in parallel with a viral infection, also called Doneth-Landsteiner biphasic hemolysin. I've also found out in my research that this isn't just viral infection. It can also be in spherocyte bacterial infections. You know, the squiggly, looks like a nail. Yeah. Now, not to be confused with enteric bacteria, but since syphilis is so easily cured now, it's more commonly found in viral infections. Auto-anti-P is a cold reactive IgG antibody and can be detected by the Doneth-Landsteiner test. So let's talk about the Doneth-Landsteiner test. Antibodies will bind to the red blood cells in vitro at colder temperatures, and when warmed up to body temperature, 37 degrees Celsius, they will lyse the cells. And that can confirm that there is a Doneth-Landsteiner biphasic hemolysin. It doesn't mean that it's going to be auto-anti-P. There are other things it can also be. I want to bring focus to just the first tube and second tube. So the first tube and second tube have the same contents in them, which is the patient's red blood cells and patient's serum. However, tube 1 was brought to 0 degrees Celsius and then brought back to body temperature, whereas 2 was strictly kept at body temperature. And you see that the red blood cells begin to settle at the bottom. But in tube 1, there is strong hemolysis. I also want to kind of just review some of the antigens and possible antibodies. I know Catie already went over some of them, but I just kind of want to retouch on some of them. For example, if you were just the P1 phenotype, then you're going to have the P1P and PK antigens present with no possible allo-antibodies belonging to the P blood group. If you are P1 negative, you're going to be having the P and PK antigens present. You will also be having the anti-P1 allo-antibodies. And we just talked about this one, P0, no antigens present, and you're going to have all possible allo-antibodies, anti-P, P1, and PK. If you are P1K, then you're going to have the P1 antigen, the PK antigen, and you're only going to present the anti-P allo-antibody. Thank you for watching.